And I think we're good to go. So I'm pleased to introduce Dr. Kimberly Cuevas, who's an associate professor of psychological science at the University of Connecticut. Her work examines the development of basic and higher order cognitive skills from infancy through childhood, with a focus on how brain maturation and underlying brain behavior associations contribute to early cognitive processes. Today, she'll be presenting a talk titled Insights into the Infant Mind, from Cognitive Processes to Neural Oscillatory Rhythms. Please join me in welcoming Dr. Cuevas.
Hi, everyone. Thank you all for taking time out of your schedules to hear a little bit about my work. So yeah, let's dive into it here. I look forward to hearing your questions and ideas. So as mentioned, I am interested in the fundamental building blocks of early cognition. And to do this, my program of research takes both a developmental psychobiological perspective as well as a cognitive neuroscience perspective. So I emphasize a lot of connections between the animal and human literatures in my work. And to do this, I look at basically three main themes within my research. One is experimental investigations of early learning and memory, which I'll speak about a lot today. Two, I look at longitudinal and individual differences analysis of emerging executive functions, and I'll talk about some of that work with a focus on EEG. And finally, I look at the neuroscience foundations of social cognition, which I'll talk about a little bit at the end of the talk. So getting into early learning and memory, I'm just fascinated about this system. The nature of infant memory has long been of interest to scientists and the broader public. And although now it seems common knowledge that young infants learn and remember, and that our early experiences have long-term consequences, as we attempt to retrieve our own earliest memories, those chapters of our lives seem mostly blank. So by no means do I have the answers to all of these questions here today, but those are the types of things that spark joy in my life. My work has examined enduring memories of infants as young as 3 months of age, as well as looking at the emergence of episodic memory. Now, when I started my trajectory of going into developmental science, I went to work with Carolyn Rovey Collier at Rutgers University as a graduate student. She's a pioneer in the field. Her work using operant paradigms provided evidence of long-term memories in young infants when the field had thought that these memories were fleeting, maybe lasting on order of minutes to seconds. My graduate student, Kelsey Davidson, and I were recently co-authoring the infant memory chapter for the latest edition of this book. And we started the chapter with a quote from Carolyn in the previous edition that I co-authored with her as a graduate student. And I really think this kind of sums up the field nicely. So, infants from their inception have robust capacities to learn and remember. And in the development of these capacities are hidden the secrets of adult learning and memory. And so from my perspective, because infants only remember for relatively brief durations, like days, weeks, and months, as compared to years, we consider infants ideal for asking some broader questions about the basic properties of memory and those building blocks for the advanced memories that we have today. So what are the methods that we use to look at infant memory? Well, the bulk of the findings on infant memory have come from three types of paradigms: preferential looking, imitation, and operant. And my work has used the latter two. So that's what I'll talk about a little bit today in more depth. So we know that babies learn a lot by doing, especially the contingencies in their surround, whether it's dyadic interactions with a caregiver or when interacting with objects. Anyone who's accidentally fallen into that game of baby drop it so you can pick it up knows that this is true. So how do we look at this in a scientific sense? So using the operant mobile paradigm, what we do is we tie a ribbon from the infant's foot and we attach it to a mobile stand. So in our non-reinforcement phase, we just look to see how much the infant kicks in general when there's a new mobile present in their crib. So you'll see this baby here, that one foot is tied to a ribbon, and that ribbon is attached to a stand that's different than this new mobile. So now we first get this rate of kicking during a non-reinforcement period. During our reinforcement or our acquisition phase, this is where learning is occurring. That ribbon is now attached to the mobile, so foot kicks will move it. And it's a type of reinforcement called conjugate reinforcement, which just means that the level of the reinforcer, how much that mobile moves and they hear the jingle bells, is contingent or directly related to the magnitude of the response, how big they kick. Bigger kicks means more mobile movement. And I have a brief video here just to give you an idea of what that looks like. So full disclosure, that is my oldest daughter who turned 5 today. That wasn't an experiment. That was just me being a scientist playing around with my baby and doing these things at home. But you get the gist of how the task works. Now, what we do is we have a learning criterion. So for a baby to have acquired this task, we look for them to increase their rate of kicking from the baseline period at least 1.5 times their operant level, or just their standard level of kicking, for 2 out of 3 consecutive minutes. Let me think, there's one other thing I want to say. Oh yeah, and when we test them, we also test them during a non-reinforcement phase. So again, the foot is not attached to the mobile, but it's attached to a separate stand, and we look to see how much they kick as a measure of memory. Now this is used with infants 2 to 6 months of age. As they get older, we have a setup like this because babies start to roll over, and that would not work with the mobile task. So we have a pack and play and a bouncy seat for them to sit in. And then you'll see that we have these colorful cloth liners around that. And we've used that to look to see how babies are encoding their environmental surround while they're learning. And indeed, we find that they are picking up on what's around them, whether it's the room or these liners. And then as an upward extension of this task for babies 6 months, and in fact, I saw a recent paper using a version of this with 4-year-olds, we use the operant train task, and it really should remind you of like a Skinner box. It is a hyped up Skinner box for babies. They press a lever, but instead of getting food pellets, they get to see this train move around the track. The train moves for 1 to 2 seconds for every lever press. And then we've got a bunch of exciting, I call it junk toys inside, like things from Happy Meals and things that light up that are all contingent on the lever press. And so this is a way to keep the infant who is now no longer laying on their back, but sitting up and using their hands pressing. But all of the parameters are the same as the mobile task. There's a baseline period, there's a reinforcement period, as well as a testing period as well. So that's some of the basic operant tasks that we have used to study memory. So when we talk about these findings, you have a little bit of background on what we're talking about. And then we know that infants don't only learn by doing, but they pick up a lot of information by just observing others. And so object-based imitation tasks in which infants observe on a single occasion target actions that are modeled with novel objects has been used in the field to examine their memories. So here is an example of a baby during a demonstration phase in our puppet task. So this baby is 9 months, and she saw the experimenter complete 3 target actions, removing the mitten from the puppet, shaking it to ring the bell, and then replacing it. And she saw that for 30 seconds. And here is another infant during the test period. So after a delay, we give infants about a minute or 2 to interact with the object. And the number of target actions reproduced within this pre-specified time is used as our indicator of memory.
That was so good.
So that baby would have received a score of 2, 1 for removing the mitten and 2 for shaking it. As you'll notice, you didn't hear any bell ring, and that's because we removed that. So it's not a cue or drawing attention to the mitten at the time of test. From separate groups, we know that the spontaneous production rate of the target actions is very low across the first 2 years of life with these acts. And we've also used within subject comparisons as well. And both of them have found really low levels of spontaneous production. So we can attribute these at the group level as being related to their memory and their learning. Okay. So based on the 3 main tasks, and I have a picture here of the visual paired comparison task as a measure using preferential looking, they have actually found a lot of similar things about the characteristics of infant memory. So one thing that we see during development is changes in encoding speed. Specifically, longer durations of exposure are required for younger infants to reach the performance level similar to their older counterparts. So they require more demonstrations of modeled actions, as well as longer familiarization and operant training phases. Another consistent finding has been the duration of memory. A lot of research examines developmental differences in how long infants remember. But one of the challenges is to investigate the specific memory process that you're interested in, retention, while controlling for others, such as encoding. So there's a lot of different techniques that have been used to equate initial coding levels across age, like having a comparable level of performance by using a learning criteria, or taking into account your immediate retention. However, under these conditions, young infants typically forget more rapidly than older infants, and that's what we see across paradigms. And I've got a graph here from a recent chapter that I think displays this in a way that is just easier to see, to visualize. So on the bottom of the graph, we have the infant's age on the horizontal axis from 2 months up to 24 months. And right here in the filled circles, this is what we see. Each circle represents a different group of infants. And these are the groups of infants that were remembered the longest. So infants were tested at shorter and longer delays. And this was the maximum retention that we saw with these standard procedures between 2 and 18 months of age. Now, this operant training task typically has 2 days of training. And at 6 months of age, infants are old enough to be tested on both tasks. And what we see is whether they're tested with the mobile task, if they learn that one, or if they're tested with the trained one, they remember it for 2 weeks. That's kind of something standard that we see with that. Okay, now for comparison's sake, if we look at the other 2 tasks, the deferred imitation task and the visual recognition memory task, we also see between 6 to 24 months of age increases in the duration of their memory. We see differences in absolute terms of how long they remember these tasks. And even though it doesn't look like a lot, from 6 months, the visual recognition memory goes from about only remembering it immediately to 24 months, it's remembered a couple days. So that line is going up. And some of the differences here are related to the parameters of encoding and training. So just as a brief example here, I added 2 more lines. The circles with the open circles represent the long-term retention for the operant task with only 1 day of training. And the deferred imitation task is seen there at 6 months with the filled diamond, that shows you with 2 days of training instead of the typical 1 day in the open fill. And so I think at 6 months, it's a really good indicator here just to remind ourselves that just like adults, we don't remember something for a specific period of time. So we wouldn't think an infant would just have a memory for a specific period of time at a specific age. It depends how long an infant remembers something. The same factors such as the amount of exposure, the number of times, the duration, all affect memory retention. Okay, so one other characteristic that we've seen consistently across tasks is there are increases in the flexibility in our memory and the conditions that promote retrieval across age. So across the lifespan, memory retrieval can be disrupted when the conditions such as the stimuli, the cues, the environmental surround differ from initial encoding. Whether infants generalize their responding to a novel test cue or in a new context depends on a range of factors, including experience and the retention interval. Infants exhibit age-related changes in the ranges of cues that support their memory retrieval. Essentially, older infants are more likely to demonstrate evidence of memory when the original focal cue or context has changed as compared to younger infants. So the work that I'm going to talk to you in more detail about today is looking at associative networks. Our findings with associative networks reveal potential underlying mechanisms for increases in transfer of learning. In other words, increasing the idea of having memory flexibility, as well as an increase in the duration of memories. So being able to protract relatively brief memories all through these associative networks. And so my work has looked at them in more detail to see how these mechanisms are developing in young age, and it's directly related to memory retention and memory flexibility. So one procedure that we have used to look at associative networks during infancy is sensory preconditioning. We know that infants learn an enormous amount of information by merely observing their surrounding, but what they learn remains latent until they have a response and an opportunity to express it. And so sensory preconditioning is a form of associative learning in which if infants form an association between two objects, so here we have the baby seeing these two objects together, which are two different puppets. If they've been learned a response on one of those objects, when tested with the other object that it was associated with, they will now show that response. Now, typically, it is not until 2 years of age do we see that memory flexibility. But sensory preconditioning as an associative technique allows a way for infants to take their learning and express it in novel contexts or in new situations. So it can exponentially increase the number of situations that infants can show previous learning. So this is something that I've examined systematically and specifically looking to see how there might be changes in how these associative networks are formed during the first 1.5 years of life. So in the initial study, I took this procedure of simultaneous pre-exposure where infants hung out in their high chair for 15 minutes a day for 2 days. They had snacks. I told the parents they didn't need to draw any attention to the puppets. The puppets are just there in the background. There's no reinforcement for looking at the puppets. They're just there. And what we found is that 6 and 9 month olds after this pre-exposure period would then generalize responding from one puppet to another. Infants who had unpaired pre-exposure, so saw the two puppets but didn't see them together, they didn't do this. And infants tested with a novel puppet did not generalize responding. So this suggested that they were actually forming association between the two puppets. We saw this again at 18 months, but surprisingly, 12 and 15-month-olds did not. When thinking about differences between infants in this age range, we thought that one of the factors could be the development of walking and how infants were becoming independent locomotive, agents, and they're seeing things more sequentially than simultaneously. They're going from one room to another. So maybe we're going to see differences in the types of pre-exposure regimen that would be effective at these older ages. So to test that out, we thought, well, what if we do the same thing, but instead of having the puppets sit side by side, what if they're presented sequentially? So they see one puppet, there's an inner stimulus interval, so there's a break, and then they see the other one. And we would manipulate this systematically to increase the inner stimulus interval to see how long of a gap could infants withstand between the two puppets and still associate them. And I should say on the side, this was all based upon work with rat pups and looking at their associations with sensory preconditioning, where they had also saw these developmental changes with the youngest pups only associating things that were presented simultaneously, and the older pups associating them when they were presented sequentially. So here So here's the graph of my data. These are imitation scores on the vertical axis that go from 0 to 3, the number of actions that they reproduced. On the left side of the graph, these are the 6-month-olds. On the right side are the 9-month-olds. And the individual bars represent which group they were in. So infants in the black bars are in the simultaneous group. Infants in the darker gray bar are in the 0-second group. And then we go on and we add longer delays. And so essentially what we saw is our 6-month-olds only associated the puppets when they were presented simultaneously, but not sequentially. And at 9 months, our infants associated the puppets whether they were presented simultaneously or sequentially. But those puppets had to occur immediately, one after the other. With longer delays, they didn't show evidence of an association. And we also saw, I won't show all of the data here, but we also saw sequential pre-exposure was effective at older ages as well. So the next question we had in understanding the sensory preconditioning process was just how quickly are young infants forming these associations? And I was particularly interested in these 6- and 9-month-old infants. So what we did is we came back to a new group of participants, and we only exposed them to the puppets one time. And the question was, how quickly could we do it? So each of these bars represent different groups of infants, and we only tested them with an exposure that was effective in that previous study. And as you'll see here on the left, with our 6-month-olds, we found that it took just under 2 minutes. That was the shortest amount of time of just seeing those puppets together on a single occasion, and then they associated them and would transfer learning from one to the other. But a minute wasn't long enough. On the other hand, with our older infants, 9 months, one time was not sufficient for either of the pre-exposure regimens. And we got to thinking that maybe what we're seeing are some changes in how infants are associating things in their environment as a function of their developmental niche or their environment of how they're learning about the world and how they're experiencing it. So younger infants don't always do things more poorly than older infants. That here's an occasion where we see that younger infants are a superior learner. They're learning these associations more quickly than older infants. But you can also think of older infants of having more tools in their tool bag. They have a lot of other ways that they're picking up information in the world around them. So to kind of get at this question some more, we also ask questions about, well, how long would a 6-month-old even remember these associations? So in each of these groups of data here, we use different types of pre-exposure regimens, how many days they were presented, how long they were presented. But the short story of it is we found that these are really long-lasting associations, that they were remembering these things for between 3 and 21 days, where they would learn something with one object, and then they would show that learning with the other one. And I don't have the data presented here, but at some of these points, they were remembering these associations longer than their older counterparts. So we saw that the 6-month-olds were actually remembering these longer than the 9-month-olds. And then another question we had was, well, how extensive are these associative networks, right? Right now, we're only looking at 2 objects, but how many things could babies kind of link together in an associative network so that one thing they could learn about and transfer that to another? So in another procedure, another study, what we did was we tried to form a 3-item puppet chain. So on day 1, they would see puppets A and B together. On 2, they would see puppets B and C together. And the question is, if we showed them something on puppet A and we brought puppet C back, would they transfer learning from one of those puppets to the other? And in fact, as you might guess, my 6-month-olds, they did do that. The bars on the left, the gray bars, show whether their demonstration, the modeling of the actions was on puppet A or C, they transferred to the opposite puppet. And the various control groups to the right, none of those groups did it. Whether they saw all the puppets but never saw them paired together, or they saw some of the puppets separately on different occasions. So then our next question was like, okay, they can do 3 puppets, but can they do 4? And just as a reminder, here is our pool of puppets. And so now we've got 3 days of seeing puppets together. And the short story is, yes, they could. And we said, well, at this point, maybe they're just forming a furry stuffed animal category. But this last bar here suggests that they're not. So that when tested with puppet E, a brand new puppet that they never saw before, they did not generalize responding to it. So yeah, we are just blown away about how these associative networks are working as early as 6 months of age. And although we presently assess the formation of just a few associations, we think that early in development, infants are readily forming numerous associations in this same way every day. And we could see how this would benefit transfer of learning. So the next thing we were interested in is this idea of memory potentiation, or the idea that a short-lived memory could be prolonged by its association with a longer-lived memory. So to do this, previous research in our lab had looked at this with 2 different tasks at 6 months of age. They looked at our puppet task, the deferred imitation task, which at 6 months, infants usually remember this for a day. And our operant task, the train task, which at 6 months, they typically remember for 2 weeks. So at the end of operant training, we modeled the target actions on the puppet. And our question was, how long would they remember the puppet if it was associated with the train? And we found evidence of memory potentiation. In other words, these 6-month-olds now remembered the puppet for 2 weeks. They would show evidence of memory. They would reproduce those target actions after a delay 14 times longer than they typically remember it if it was associated with this train, which they remember for 2 weeks. The same additional groups of infants in these procedures were tested after 3 weeks. When they don't remember the train, they also don't show evidence of memory of the puppet. So if that isn't cool enough, we wanted to take this even a step further to see how complex these associative networks are. And so what we did is we said, well, if infants associate 2 things that they see together, what if they just think about them, even if they're not physically present? So if a baby is thinking about an operant task like our mobile task, and they're thinking about our puppet task, like, would they associate them if they're just thinking about them and they're not physically present? Okay, so I know this sounds crazy, like how would we do this experimentally? So that's what I'm going to show you on the next slide. And as I tell you, I take a developmental psychobiological perspective. So we were reading an animal rat study with adult rats, and they had done this sort of idea of associatively activating event representations. And I'll walk you through that real quick and then tell you how we did this with infants. So with rats, in phase 1, they were exposed to a peppermint flavored solution in a distinctive context. In the next phase, they were exposed to an almond flavored solution that also had sucrose, and rats like sucrose. So phase 3 was the key phase. They presented that almond flavor within context 1. And what they thought would happen is having context 1 present would retrieve the memory of peppermint, even though that isn't present, and tasting almond would retrieve the memory of sucrose, even though that wasn't present. So if now peppermint and sucrose are coactive in memory, although neither of them are there, then later rats would show a preference for peppermint as compared to another solution, and this worked. And so we said, if rats can do this, we think 6-month-olds can do this too. But we're not doing solutions. We're using these battery of tasks I've already told you about. So in phase 1, we use our sensory preconditioning technique to form an association between 2 puppets. We have a puppet A and puppet B association. In phase 2, infants learn to kick the mobile in a distinctive context. So now we have a mobile context association. In phase 3, we place puppet A in context 1. So now at this point, puppet A should retrieve the memory of puppet B that was formed in phase 1, and the context should retrieve the memory of the mobile that was formed in phase 2. So even though neither of these were physically present, we predicted that they would be associated. Now, how would we know if infants associated them? The next day, we had a novel experimenter come and demonstrate the target actions on puppet B, the one that they haven't seen since phase 1. And we said, how long will they remember the puppet? If they remember it the same duration as the mobile, then that will give us evidence that they were associated, even though neither one was physically present. So these are the forgetting functions that I've already showed you just for reference of how long they remember the mobile and the train. So now I'm going to click on here, our novel group. So the open circle is our associatively activated memories of the puppet and the mobile. And what you see is these are all separate groups of infants. Infants remember the puppet now, puppet B, for 2 weeks, the same duration that they remembered the mobile, but not 3 weeks. And various control groups with separate exposure did not. So memory potentiation through associative networks is yet another way infants are expanding their network. They're expanding how long they typically remember items. And crazy enough, they can do it by just thinking about these things at the same time. I bring us back to this figure about the duration just to say that there's been subsequent studies that have looked at memory potentiation with not just using the operant task, but looking over here at these 2 other tasks, the deferred imitation task and the visual recognition memory task. This subsequent research has found that visual recognition memory can be extended by being associated with the longer deferred imitation memory. So for example, one study looked at 24-month-olds and saw that 24-month-olds would remember the visual recognition memory if their encoding occurred in the presence of the puppet in which they saw the target action demonstrated. So now they wouldn't remember this visual recognition preference for a matter of days. They'd remember it for 8 weeks, the same duration that they remember the puppet. So I think that these things are all really amazing and showing how the early associative network is extending our memories and also extending the situations in which we show long-term memory. Today, I focused in on just a brief section of the work that I do on associative networks and contingency learning, but I also have a line of work of looking at episodic memory with older kids at 3 and 4 years of age. And what we've done in those tasks is to look at their memories, both verbally as well as behaviorally. And we've done some work looking at that longitudinally. And what I hope to do in the longer term part of my career is to really kind of connect those two fields, is to connect what we see during infancy and how these are related to the emergence of episodic memory abilities that we typically talk about in terms of 3 to 4 years of age. Okay. So I realize I've used up most of my time here talking about early learning and memory. So I am going to skip through to talk about just a few things about infant EEG. We also look at infants' neural activity to see how that is related to their ongoing cognitive processes. And 2 lines of work that I have done this with is looking at infants' executive function. So specifically looking at their working memory and inhibitory control, as well as looking at their action perception abilities. And my 2 graduate students, Lauren Bryant and Kelsey Davidson, have been essential in this work. So EEG is ideal for use in younger infants. It has optimal temporal resolution, it's non-invasive, and it's relatively resistant to motor artifacts, and it's inexpensive as compared to other imaging techniques. What we do know is that there are changes in the EEG neural oscillatory activity as a function of age. So you can break up the EEG oscillations into different frequency bands. And each of these frequency bands, although you might typically hear about it in the context of sleep, we do know that they are related to different cognitive and perceptual processes in the awake EEG. So the measure that I talk about most often is power, which is a measure of the excitability of groups of neurons. Now this tracing, one is from frontal sites in an infant and one is from frontal sites in adults, is just to show you how different the EEG signal looks as a function of age. And these are both during periods of quiet wakefulness. When we look at the longitudinal spectral plots, we know that as a function of age that there's changes in the peak frequency of the EEG, as well as changes in the distribution of power. And the reason that I mention this is that most of our work focuses on the infant and child 6 to 9 hertz EEG. It's also been called the infant and child alpha rhythm. This is thought to be analogous to the adult 8 to 13 hertz alpha rhythm. And this rhythm has been looked at in terms of executive functions, action perception processes, as well as affective processes. And my line of work has looked at it within the context of executive function and action perception processes. Now, we know that executive functions are higher order self-regulatory processes involved in coordinating, planning, and completing goal-directed actions. In infancy, we typically look at working memory and inhibitory control. And we know that the prefrontal cortex is playing a large role in executive processing. So the task that we have used to look at this is the delayed response task, or from your developmental psychology text, you can think of it as the A not B task. This is a simple hiding task in which the time between where the item is hidden and when the infant searches for the item is used to look at their working memory and inhibitory control. Martha Ann Bell has developed a looking version of this task that can be used for EEG. And so that is the measure that we have used here in the following slides. But we have done work longitudinally comparing within subjects children's performance on both tasks. And we see that once infants can reach that there's no differences in their performance as a function of modality of the task. So just as a quick picture here, so we have non-reversal trials where the item is hidden on the same side as a previous trial. We also have reversal trials where the hiding location is switched. These trials require additional inhibitory processes. We know that there's individual differences in infants' working memory and inhibitory control. So we use a progressively more challenging design. Essentially, if infants are able to find the toy successfully with a 0 brief delay, we then increase the delay incrementally by 2 seconds until we find a delay in which the infant starts making the error. So just to orient you, the portion again of the EEG that we're talking about is after the item is hidden and before the infant looks for the item. And we're going to look at their brain activity during that brief delay. So what we found is at 5 and 10 months across the scalp, that during that brief delay, infants are showing increases in EEG power. However, if we look at individual differences, it's the magnitude of their frontal EEG power that's associated with their performance on the task. Next, we looked at task demands. So we compared their EEG during that working memory component to same side non-reversal trials to the reversal trials that have the additional inhibitory demands. And what we used is a measure of functional connectivity or coherence. So how much synchrony do we see across the scalp? And what we see is for those increased inhibitory demand trials, those reversal trials, we see more frontal functional connectivity. And finally, we could compare trials that infants were subsequently correct or incorrect. And we looked at functional connectivity once again. And what we found is for the trials that they were subsequently correct, that they showed increases in frontal functional connectivity. We've done a lot more work on this, and this is just to show that measures of the EEG are associated with infants' performance, both concurrently as well as looking at their future executive functions. We performed a recent meta-analysis looking at self-regulation and frontal alpha power, and we did see associations that were stronger when infants were older, aka when they were preschooler, these associations were stronger. When these were measured by behavioral tasks as compared to like parent report or questionnaires, as well as when we looked at measures of executive function as compared to other measures of self-regulation or emotion regulation. And so this is a long way to say that we found evidence that executive processes are associated with infant and child frontal alpha measures, both functional connectivity and neural activity, in these rudimentary forms of working memory and inhibitory control during infancy, even though we know this is very early in the maturation of the frontal cortex. And just as an aside, what I find really interesting is Nick Turk-Brown out of Yale has recently published papers where he's using fMRI and doing imaging of awake infants. And in a recent task of looking at attention processes, he has found evidence of frontal activity during attention processes in infants as young as 3 months of age. So this is really exciting to see some converging evidence of, even though we know that the frontal cortex is so immature at this age, that in my opinion, these experiences during infancy are helping it reach functional maturity at the level that we think of later in life. So on that spot, I am going to wrap up and not show all of these other slides, but I can't not skip through them. And thank you all for your time. I would like to thank the graduate and undergraduate students, the families, as well as the support from UConn, as well as NIH for the work that I presented here today. So I look forward to talking with you all and hearing your questions and comments. Thank you.